Hi, my name is Yan Ko. I'm a PhD student at Dr. Avi Mayan's lab. In this and the following lectures, I will guide the basic analysis of next-generation sequencing step by step. I will also introduce the basic usage of Linux/Unix commands and software R, as many bioinformatics tools are built with these open-source platforms. Let's first start with the RNA seq analysis. Before we start the analysis, let's first review some simple facts about RNA seq. Why does it get popular among the research community since the past few years? What's the advantage of RNA seq over microarray? What's the limits of the RNA seq? And the topic of this lecture, what's the standard pipeline to process RNA seq data? So RNA seq is a high-throughput or next-generation sequencing method to measure the genome-wide transcriptome or RNA content of a given sample. The RNA is extracted from the cell, reverse transcribed to cDNA, and chopped to short sequence, followed by massive parallel sequencing. The sequences are generated as short reads and will be mapped to the reference genome if there are any, or de novo assembled to the transcriptome if the reference genome is not available. So transcriptome sequencing, aka RNA seq, can evaluate absolute transcript levels of sequenced and unsequenced organisms, detect novel transcripts and isoforms, identify previously annotated 5' and 3' cDNA ends, map exon-intron boundaries, review sequence variation and splice variants, and many more. During the past 5 years, next-generation sequencing was established for almost all DNA-based molecular research field and therefore could be considered as an all-in-one platform. As more data gets deposited in the public domains, meta-analysis can be implementable as microarray data has been explored. However, the usefulness of RNA seq is limited by both experimental and analytical factors. The non-uniformity coverage of the genome introduced biased transcriptionally active genes. Although being improved, RNA seq is not good at capturing short-lived transcripts such as microRNAs. The sequencing error rates of next-generation technology is usually low, but as the massive amount of reads are being generated, the mapping uncertainty increases. And besides, current tools usually fail to map repetitive elements. As for the downstream data analysis, the mapping and assembling algorithm still needs to be improved, and the bioinformatics software needs to be simplified for the use of biologists. A more practical bottleneck, the economical benefits seem not to be sufficient enough. Different research groups and companies are currently working on developing the third generation of sequencing. And in the near future, for example, sequencing the human genome can be low as less than $1,000. So this is just a simplified introduction about RNA seq. If you'd like to know more, I'll list several reference reviews at the end of the slides. Now let's get down to data processing. When measuring the transcriptome, the most common task is to identify differentially expressed genes between treatment and control experimental conditions. Sometimes, the experiments are done at different time points or different dosage, and we like to know the differentially expressed genes across a series of conditions. Therefore, the example data we use in the tutorial is designed as a knockout and wild-type samples from 3 different time points. We will identify the differentially expressed genes between knockout and wild type at each time points, as well as the differentially expressed genes between the neighboring time points, using the pipeline of one of the most popular RNA seq analysis software called TopHat and Cufflinks. This is the workflow of the RNA seq analysis pipeline. Once we got the short reads from sequencer's output, the first step is to map it to the genome using TopHat. Here we're dealing with the situation where a reference genome for the organism is available and well annotated. If no reference genome is available, we will need to assemble the transcriptome using the de novo method. The details of the de novo transcriptome assembly can be found in the reference. Top hat is a sequence aligner designed specifically for RNA seq. The regular next-generation sequence aligners fail to map RNA seq reads since there are discontinuous sequences that connect two exons. Top hat breaks down these junction reads and searches for the best neighboring exons that yield those sequences. The next step is to assemble the transcriptome with cufflinks. It takes the aligned data from top hat and measures the transcription level by the normalized number of reads mapped to the defined regions, such as transcripts, gene and isoforms. Differentially expressed genes can be identified using the cuffdiff module in cufflinks. Once the transcriptome is constructed, we will be able to visualize the RNA-seq data with the R package cummerbund and proceed for further analysis, including hierarchical clustering, pathway analysis, enrichment analysis, principal component analysis, etc. I will introduce cummerbund in the following lecture, and these popular data analysis methods will be covered by the rest of this course. One important thing to bear in mind for the data analysis is that always check the quality of the data before any further analysis. For example, what's the quality score distribution of the sequences? How much of the sequences can be mapped to the genome? Are expression levels of different samples within the same magnitude? Unlike microarray, the number of replicates of our RNA-seq experiments is usually limited by the amount of biological material and economic cost. Therefore, the data analysis workflow is slightly different for the two situations. For example, for experiments without replicates, the transcriptome assembly and differentially expressed genes identification can be combined as one step using the cuffdiff functionality in cufflinks. When replicates are available, it's recommended to first assemble the transcriptome individually for each sample and then merge the replicates into one assemble afterwards. Differentially expressed genes can be then identified using the cuffdiff once the transcriptome is constructed for each biological condition. Cufflinks has provided smooth functionality to handle both tasks. Using the top hat and cufflinks pipeline is challenging for biologists since it's designed to run on the UNIX/Linux system. Although most of the popular Linux distributions come with graphic user interface, top hat and cufflinks do not have such utility, and everything will be invoked and set up from the command line. Therefore, we will first learn some basic Linux commands before we can start. The commands will be demoed in the Linux system. For Mac users, simply open the terminal and the commands will do exactly the same as shown in the lecture.
For Windows users, since the default command prompt is not Linux-based, we need to mimic the Linux environment with the software Cygwin. I've written the step-by-step instructions about how to install Cygwin in Windows. You can find it in the assignment for this lecture. First, let's open Cygwin. The default directory when you open Cygwin is the home user directory. Cygwin treats the file system differently as Windows. It puts all disks under one folder called C drive. I will show it later. To find out the current directory, we can use the command pwd. Next, we want to see what's in the directory, and we use the ls command. Right now it's empty, so nothing shows up. Now let's go back to the root directory and take a look at the file organization. We can use the cd command, short for change directory, to move around. You can also type the path to which you want to move to, or use the .. to move one level up. Here I use twice to move up to the root. List again, and we can see the root folder content. It's similar to the Linux system, but not exactly the same. I mentioned that all disks of the Windows system is put under the C drive folder. Let's get in there and take a look. List again, we can see two disks. C is the default drive and Q is my external hard drive. Move to C drive and list all files. Now we can see something similar. We have the documents and settings, users, program files, etc. The example I want to show is in document folder. So cd to there. Oh, here it is, the tutorial folder. I'll put a text file in this folder. It's actually the gene annotation of the mouse genome. We'll touch it later. First, let's do some simple operations. To create a new directory, we can use mkdir command followed by the name of the new directory. For a new dir, for example. Now list it again and we can see the new directory we just created. Of course it's empty because we haven't put anything in there. Let's go back and copy the text file to the new folder. The Windows style copy and paste can be done by using one command, cp. Simply type cp followed by the name of the file we want to copy and the path to the directory, and it's done. The slash means this is a directory rather than a file. Go to the new dir and it's already pasted. Well, do you wonder what about cut and paste? In Linux, the cut and paste operation is combined with the rename of file, meaning that these two share the same command, mv. For example, if we type the file name followed by a new file name, the file will be renamed. But if we type a directory instead of new file, the file will be cut and paste to the directory and the original name of the file will be kept. Here we can see this file is renamed. Next, I want to show you how to view the top or bottom of a file. This is particularly useful to deal with genomic files since they're usually very large and regular text editor either won't open them or dramatically slows down your computer. But genomics files usually contain structured data. Therefore, we can view only the first few lines or last few lines to get a sense of what the data looks like without opening the entire file. The commands are head or tail. Using head followed by how many lines you want to see, followed by the name of the file, we will get the first few lines shown in the terminal. For example, here I want to see the first 5 lines of this mouse genome file. It's a bit hard to tell how many lines are here since they're wrapped, but if we count the genes, we can see it's 5. Same as head when we're using tail. We're showing the bottom 5 lines of the text file. One of my favorite Linux commands when dealing with large files is a command that counts how many lines are there in a file. You remember how annoying it is to open a large table in Excel and drag your mouse down to the end just to see how many rows are there? Well, in Linux, you can just type wc-l followed by the file name and the number of lines will show up instantly. Here we have 30,000 something transcripts in the mouse genome. The last thing I want to show is the grep command. It's extremely handy to handle simple text files. For example, in this genome annotation file, each line contains a transcript. Therefore, for a single gene, multiple lines exist if it has more than one transcript. Now I want to see all transcripts for BRCA1. I can use the grep command to extract all lines and match the string BRCA1. It turns out BRCA1 has only one transcript. Using grep, we don't need to give the full name of the gene. For example, I want to see all transcripts of genes that start with FUT. I can type grep FUT, no numbers, and I'll get a full list of genes beginning with FUT. As I mentioned, grep has a lot of parameters we can play with. Where to find them? As a matter of fact, most Linux commands come with a set of parameters. To use them, we need to read the documentation about the commands. Fortunately, this can be done in the terminal. This is what Linux guru called man page. Simply type man followed by the name of the command, and the full documentation about the usage will show up in the terminal. Name, synopsis, description, etc. You can type Q to quit the man page and get back to the terminal.
So to remind you, this is a screenshot of the commands. And if you're still cloudy about these user-unfriendly commands, get a new lab suite like this, which has printed all essentials upside down. The last prep before we start a pipeline is about the file formats. As we know, sequencing data are very large, therefore it's stored in specific formats. The most frequently used format is FASTQ. For each short read, 4 lines of information is written in the FASTQ file. The first line starts with @ symbol, is the sequence ID. It's usually generated by the machine. And the second line contains the sequence of the short reads in ACTG codes. The third line starts with the plus symbol and it's optional. The fourth line contains the same number of symbols as the sequence in the second line, and it encodes the quality value of the sequence. The quality value can be read by some software to evaluate the sequencing quality. Each FASTQ file aggregates millions of these reads in the same format. Top Hat takes the FASTQ file as input, aligns each read to the genome, and generates a BAM file that contains the chromosome coordinates of each of the reads. The BAM file is a binary, compressed, and indexable representation of the alignments. Therefore, it's not human readable, but is convenient for visualization with the genome browser. Some well-established tools such as SAMtool or BAM tools can be used to work with the BAM files. Here is a list of the recommended readings. The review articles give a detailed introduction about the computational methods for next-generation sequencing analysis. The 2 other papers describes how does Top Hat and cufflinks work. In the next lecture, I will guide you through the Top Hat and cufflinks pipeline step by step. Thanks for watching. I'll see you in the next lecture.